All right. So the Hardy-Weinberg equilibrium is, or theorem, states that allele frequencies and genotypes in a population's gene pool remain constant from generation to generation. Okay. So one of the foundational things that we have to understand here is something known as allele frequencies. And that is something that I thought the POGIL kind of did pretty well. It had you look at a population, a small population of beetles, and in that population, you saw what the genotypes were, and you were able to literally count how many alleles were in the population and what versions of those alleles were in the population. And so what Hardy-Weinberg, as the theorem says, will allow us to understand, is that population of individuals, if they reproduce, will their allele frequencies be the same as their current population for their offspring? And if they are in this equilibrium, theoretically, those frequencies should never change. Now, the change in allele frequencies is also known as evolution, okay? And we have to kind of go back to the base understanding, very generic understanding of what evolution is. I've not done this, but I'm sure if you looked the word to evolve or evolution up in a dictionary, there'd probably be a really generic definition of it that states something like something changes over time, or change over time would be a very stock generic definition of evolution. Now, we as human beings, our minds, when we hear evolution, we default to this idea of monkeys turning into humans, or dinosaurs turning into birds, or dinosaurs going into reptiles, or whatever. That's what our mind ultimately goes into. But we have to recall that it's nothing more than change over time. And when we're talking about population evolution, the population could theoretically change over time as well. And it could be as soon as one generation. So we are measuring evolution at a very small scale, really, in terms of time. You can measure it over one generation. But if a population is in Hardy-Weinberg equilibrium, it means that if that population had babies, that their babies would be representative of the same allele frequencies. So that's one of the reasons I think that and we'll talk about these 5 conditions later when we get into some of the sample problems we do tomorrow. So I don't want you to get caught up in this stuff just yet. But one of the things I want to talk about and expand on and review is this idea of the allele frequencies, because it's going to be something that we continue to try and understand, and we're going to continue to use throughout the course of solving some of these problems, okay? So we have some practice with this with the POGIL that we did, right? And I think it's a really good way to practice to visualize something, okay? And to think about something abstract as allele frequencies in a more concrete manner. So I'm hoping that as I kind of go through this, thought experiment myself, or this imagination process myself, that you guys would try to do it at some point as well. So this scenario here, and I'm glad it's very black and white and stock, and there's really nothing going on here other than just a few words and numbers, is going to be something that we can visualize in our heads. And as biologists, it's very important that we learn how to visualize processes and diagrams and things like that. So try this with me, okay? So visualize, and I like to close my eyes when I do this because it helps me. Visualize a field, a beautiful field, okay? It's springtime, right? And in this field, there's these wildflowers. They're all over the place. And you've got red ones and pink ones and white ones. And you see way more reds than the others, and there's some pink intermingled in there. And then there's also some white ones that kind of add that little flair or whatever. So you've got all these flowers in this big field, right? And those flowers have a specific color because of their genes. Okay, so that's something we have to realize, because of their specific allele combinations that they have. So in other words, this field of flowers is also a giant pool of genes. Okay, that's where you get the term gene pool. And so you have to then think about the flowers now as just kind of alleles and genes. So every time you picture a flower, you should try to picture what the inside of its cells look like, and then inside the nucleus, and say, okay, I see a red flower, and I'm looking at the chromosomes, and there's one big R and another big R right next to each other. That's why it's red. And so in this field of 500 wildflowers, 320 of them are red. But we should also think about this as how many R alleles do we have? So each one of those flowers has how many big R alleles? How many of those flowers has, or each one of those flowers has how many big R alleles? 2, right? So how many total big R alleles are there in the population based on these red flowers? How many total? So if I were to go into each one of these red flowers and I count, okay, here's one, here's one. I go to another red flower, I say, here's one and here's one. How many do I add up from just the red flowers? What's that? 640, right? So because of those red flowers, there's 640 red alleles, big R alleles. Okay? Let's do the same thing with the white flowers. Okay? How many W alleles are there? How many? 40. Good, because each white flower has 2 alleles. So there's a total of 40 W alleles just from the white flowers. Now the pink flowers are a little bit different. The pink flowers have 1 big R and 1 big W. Incidentally, can anybody tell me what kind of dominance that is? Beginning of the semester stuff. What kind of dominance creates a pink flower like we see here based on that? Anybody at home got this one?
Codominance.
Oh, you're close, but no cigar. Nick? Boy, it's incomplete dominance. Incomplete dominance, where either one doesn't really know if they want to be dominant over the other, so they kind of just share the responsibility. So, how many big R alleles are there in these pink flowers? How many big R alleles based on what we see in the pink? 160, right? Because each pink flower has 1, and we have 160 pink flowers. So then how many W alleles from the pink flower? 160. Easy peasy, right? Okay, so what we've literally done here is take stock. We've counted how many alleles of each color are in all of the flowers, okay? So now let's tally them up. We have a total of 800 big R alleles, and we have a total of 200 W alleles, all right? So how do we find what's called the allele frequency? We do some dividing, okay? So we take 800 divided by the total number, which is 1,000, and we get 0.8. And then we do the same process here for the W. Get 0.2. Okay. So when we use this term allele frequency, what it does is it breaks it into a statistical thing, okay? It talks about something random, because what we can do with these frequencies is turn them into percentages, right? We can say, okay, 0.8 is actually 80%. So the way we can interpret this is now here's me imagining myself being blindfolded as I close my eyes, if you can get that. Okay, so I'm gonna close my eyes, and I'm gonna imagine myself walking through this wildflower field with a blindfold on, and I walk up to a flower. I don't know what the flower looks like, and I pick it up, and I reach in there, and I look at an allele. I reach into the flower's DNA, and I grab one of the alleles that it has. What's the chance that it's a big R? 80%. Okay, so there's an 80% chance that if I just go out in this field of wildflowers and I pick one up, 80% chance there's going to be a big R allele in it, okay? So I do the same thing, and I go out and I pick this one. I have no idea what color it is or what it looks like. What's the chance I'm going to pick out a W? 20%, right? So there's a 20% chance that I can just go pluck a W allele out of this population. And so this same idea can be applied to mating, because that's effectively how this works in natural populations. There's an 80% chance that the big R allele will be passed on to the next generation, and there's a 20% chance that one of those W alleles will be passed on to the next generation. Okay? And so by understanding these probabilities and chances, we can then kind of predict what an offspring in the next generation would look like. Okay? And so that's how we can utilize some of this data and use it to interpret what's going on in the next generation, right? And so a lot of that is stuff that we covered yesterday, but I think it's very important to understand the allele frequencies and then be able to apply them. So what we're going to do now is I'm going to introduce the Hardy-Weinberg formula and kind of describe what some of these things mean. But the reason we kind of review that is because it deals with allele frequencies and using the allele frequencies once we've figured them out, okay? So down at the bottom here, we see the Hardy-Weinberg formula. And up here, we see things introduced called P or Q. All right, so I want to talk first about what P and Q represent, okay? Just like any other math formula, somebody who came up with this formula picked letters to represent stuff, okay? And so Hardy and Weinberg came up with this formula, so they get to figure out what these things represent. And they said that P, the letter P, is going to represent the frequency of two possible alleles, and P represents the frequency of the dominant one, okay? So in our previous example, I'm just going to pick red. It's not how it really works, but we're going to say red, the R, which equaled 0.8. We're going to say that equals P. We're going to say that was the dominant one, okay? And we're going to say W, which equaled 0.2. We're going to see that equals Q, because that represents the frequency of the recessive allele. How often we would see the recessive allele in a population. That's what it's trying to tell us. So we have to get accustomed to using P and Q because they should mean something to us. Whenever we see P, think frequency of dominant allele, okay? In other words, how often do you see the dominant allele in a population? When you see Q, think frequency of the recessive allele. In other words, how often you see the recessive allele in a population, okay? What's the percent chance you pull a W out of that population, okay? And so they further go on and elaborate on this, and that's what I thought the POGIL did a really good job of doing, was explaining where we get P squared and Q squared, and also 2PQ. Whoops. And also where we get 2PQ, okay? So now P squared and Q squared also represent something, okay? But they represent something specifically different. They represent the frequency of homozygous genotypes in individuals of a population, okay? So P squared, what individuals would they represent in a population? Homozygous what? What does the P represent? Dominant, right? So P squared is going to represent the frequency of dominant individuals, homozygous dominant individuals in a population. In other words, when we calculate P squared, it should tell us what proportion of the population has that particular genotype, okay? And likewise, Q squared, when we calculate it, you're looking at Q squared, it should tell us the percentage of a population that has the homozygous recessive phenotype. So if you did jump in, for instance, if you did jump into the sample problem, I think number 21 on the POGIL, it mentioned that 9% of the population had sickle cell anemia, okay? And sickle cell anemia is homozygous recessive, okay? So the frequency of the population that has sickle cell anemia was 9%. In other words, Q squared equals 9%, Okay, so that's what it's trying to get across. That's what it's trying to help us understand is what these equal. And then 2PQ is representative of the heterozygotes in the population. How many heterozygotes in the population represent that heterozygous genotype? Okay. Now, one thing that I will mention with these is that when you start to work with them and you start to do problems with them, an understanding of P, Q, Q squared, P squared, 2PQ, all that stuff, he doesn't like it. All that stuff starts to get much more clear when we start working with it, okay? So I anticipate over the course of the next couple days as we do more and more with this stuff, that you're gonna get a better, more thorough understanding of what this kind of gobbledygook new mathematical formula is trying to tell us. And then be able to manipulate it to predict certain things, which I think is really fascinating about this formula, okay? So that'll do it for new stuff that we're gonna like learn. Now, those of us who are in person are gonna go play with cute chickens.